So question number first from chapter third is what is replication, right? So let me give you, so the question was what is replication? So let's consider this is a DNA, double helical DNA. Now, in prokaryotes, there is no nucleus, so this DNA is present in the cytosol, whereas in eukaryotes, let's say a human cell, this DNA is present in the nucleus in the form of chromosome. So we already studied the chapter on cells division, where one cell divides into two. So it is very important that when a cell is dividing, dividing, it has to duplicate DNA. So the DNA has to exactly duplicate itself before the cell division, and each daughter cell should get equal number of chromosome or DNA. So let's say if this DNA is duplicating, then this should give rise to two daughter DNA molecules. Two daughter DNA molecules, right? And each daughter DNA molecule should be passed on to each daughter cells. Now this process of DNA duplication is called as replication. Now this replication is carried out by an enzyme called as DNA polymerase. Right? So this is what the question refers to. What is DNA replication? Now I would like to add here that in bacteria, replication occurs in cytosol. Why? Because bacteria has no nucleus, whereas a human cell or a plant cell, right, or a plant cell, which are eukaryotes, they have nucleus. Replication occurs within nucleus. Now, what is the importance of replication? Is of course duplication, but the core value of replication is that the quality of DNA should be maintained from one cell. When it is dividing into two cells, the quality of DNA should be maintained. If it is not maintained, then it can give rise to serious diseases in humans, especially cancers. So so how does replication happens? So let's say hypothetically that this is a DNA molecule. This is a DNA molecule, right? Then each one of the DNA molecules, each of the DNA strands, so DNA is made up of two strands, it's a double helical strand. So each of these strands will give rise to, a daughter strand. So fidelity, the loyalty has to be guaranteed, because even if there is a single change in a nucleotide, then it can give rise to serious mutation. So this is essentially the basic of replication. Okay. So what's the next question? The next question is, what is transcription? So here we spoke about replication. So this is one of the functions of DNA. The second function of DNA is that that DNA has to control our cells, the cell functions, cell division. DNA has to control many functions. DNA cannot step outside of the nucleus and do all the functions. So DNA is very critical. Why it is critical? Because it is a molecule of inheritance, and so it has to be maintained. The quality of DNA has to be maintained from one generation, and I'm not talking about one generation, but millions of or hundreds of generations. The quality of chromosome and DNA has to be maintained. And one of the ways it is maintained is that DNA is housed in the nucleus. The chromosomes are present in the nucleus. It cannot go everywhere. But DNA has the message through which it can pass its orders, its message. It can convey through series of steps. And what are those steps? So there are two steps by which DNA can do. And these steps, they follow one another. So first is transcription, and transcription is followed by translation. Now, so DNA essentially could start the process of transcription, and then transcription could lead to translation. So what is transcription? So transcription is a process by which the same DNA can work to synthesize a new type of molecule called as RNA. And this, when DNA serves as a template for synthesizing RNA, this process is called as transcription. So this process is called as transcription. Now, RNA is again very sophisticated molecule, very labile. It is very, very delicate. So RNA then again serves as a template for the synthesis of another molecule, which is called as proteins. So I revise. DNA does two functions. One, replication, and the second is transcription, and the end product of transcription is translation. And what does translation means? Translation means synthesis of new proteins. Synthesis of proteins. That's what translation. That's what happens in translation. Synthesis of proteins. Now, what did I mention to you? That in bacteria, the process of replication occurs in cytosol. Why? Because there is no nucleus in bacteria. In humans, the process of replication occurs in nucleus. Same is the case with transcription. So in both the situations, in bacteria, in both bacteria and human nucleus, transcription also occurs in nucleus. Transcription also occurs in nucleus. So I repeat. So in bacteria, in bacteria, replication transcription both occurs in cytosol. Why? Because there is no nucleus. And in plants and human cells, so let's say in eukaryotes, both replication and transcription, they occur in nucleus. Why? Because the DNA is present in nucleus. So transcription and replication in eukaryotes, both in nucleus. In bacteria, it is in cytosol. Now, this is a very important point. Point is that translation in both bacteria as well as a human cell or a plant cell, translation occurs in cytosol. Why in cytosol? Because the primary site for protein synthesis is ribosomes, and these ribosomes, they are present in cytosol or cytoplasm. So what is the message for this whole slide is that DNA is engaged in two processes, two important processes, important in the sense for the survival of the cells, because if there are no proteins, there are no essentially workforce within the cell or a body to do the functions. So cells will not survive. And if the cells want to divide and grow, but there is no replication, again, the system will collapse, the living cell will die. So two important functions of DNA, replication and transcription. Replication is carried out by an enzyme called as DNA polymerase. Transcription is carried out by an enzyme called as RNA polymerase. And so these are the differences between the replication and transcription in prokaryotes and eukaryotes. And translation is a process where RNA serves as a template for the synthesis of proteins. Okay, so let's go to the third question. So third question is, what are the difference between replication and transcription? So I just mentioned to you, I answered this in this just now, that replication is a process where DNA is duplicated by an enzyme, DNA polymerase. And transcription is a process by which DNA serves as a template for the synthesis of RNA by an enzyme. This is carried out by an enzyme called as RNA polymerase. Fourth question is, what is translation? I just mentioned to you just right now. The translation is a process where RNA serves as a template for synthesis of protein. So what's the fifth question? The fifth question is, what are the roles of mRNA, rRNA, and tRNA during translation? So translation is a very sophisticated process. It has to be done meticulously because it involves many things. It involves DNA, it involves ribosomes, and it is an assembly of one of the major macromolecule called as proteins, and proteins are workforce. So this is one of the most important processes that a cell is involved. So what cell does, cell is very smart, and what the cell does is, is division of labor. So it gives duty to various molecules, and these molecules, they do a teamwork, and when they do the teamwork, they lead to the synthesis of proteins. So let me tell you this, why I made this connection is that DNA serves as a most important body of a cell. This DNA serves as a template to make RNA, and that step is called as transcription. And then RNA serves as a template for the synthesis of proteins, proteins through process of translation. Now, I just mentioned to you that DNA is always present in a human cells, always in the nucleus. Cytoplasm is always, RNA, or let's say the translational machinery, which is called as ribosome, is always present in the cytosol. So DNA is present in the nucleus, ribosome in the cytoplasm. There has to be some messenger. That messenger is your RNA. But this process of protein synthesis is not simple. It needs more molecule, it needs more partnership. So what did nature do is that nature made 3 kinds of RNA. Not 1, not 2, but nature made 3 kinds of RNA. Now, what were those RNAs? Those RNA, one of them was mRNA, right? And as the name suggests, M means messenger. The second kind of RNA was tRNA, and as the name suggests, this was transfer RNA. Transfer RNA, as the name suggests. And the third kind of RNA is rRNA, where R stands for ribosomal RNA. Right? So what happened? I said division of labor, it needs more. So to run a company, a person cannot run company by him or herself. They need a team, they need a collaboration, they need a structure. So cells DNA is very important. So it cannot do all the functions. It needs partnership. It needs more. It needs to employ more people. So what it does, it counts on RNA to have 3 types of RNA: mRNA, tRNA, and ribosomal RNA. Now, so what was the question? How many types of RNAs are produced during transcription? So three types. There are more types, but at this stage, you need to know only 3: mRNA, tRNA, and rRNA. So certainly if there are 3 types, there are 3 different types, so means they're having 3 different functions, right? So what are their functions? So this mRNA, it serves as a template for amino acids. What is proteins? Proteins are, from the chapter 1, proteins are macromolecules. And what are the building blocks for proteins? It is amino acids. How many amino acids are there in our cell? At least 20. Right? So 20 amino acids. So how do you know which amino acid, right, has to come to make a protein? That code is present in messenger RNA. So it serves as a template. The second type of RNA is transfer RNA, and the function of transfer RNA is that this transfer RNA brings amino acid to the ribosome. Now you have to think that ribosome is present in cytosol. The translation has to occur on ribosome surface. And what is translation? It is protein synthesis, amino acid. So who will bring the amino acid, right? So the delivery person is tRNA. So mRNA serves as a template. tRNA brings the amino acids. So there is a code that says, okay, this is the sequence of mRNA. So this protein should come. This amino acid should come. Now, if several amino acids came to make a protein, who will make the peptide bond? So the peptide bond between amino acid is made by ribosomal RNA. Peptide bond. peptide bonds, right? So transcription and translation is a teamwork that is orchestrated by, of course, the starting point is DNA. It is orchestrated by enzyme RNA polymerase that synthesizes RNA. And RNA, they need to do different functions. So there are 3 types of RNA: mRNA, tRNA, and ribosomal RNA, rRNA. And then these 3, they come together to synthesize proteins, and all these happens at the surface of ribosome present on the in the cytosol. So that's why transcription in eukaryote cells happens in the nucleus, but RNA, they have to exit, they have to shuttle from the nucleus into the cytosol and reach out to ribosome on the surface, and then get the protein synthesis operational. So let's look at, what's this next question? Yeah, what is the function of ribosomes? So the function of ribosome, as I mentioned, ribosome serves as a platform for the synthesis of proteins. So it serves as a platform, but who are the actors? So ribosomes, it serves as a stage, but who are the actors for protein synthesis? mRNA, that serves as a template. tRNA, that brings amino acid. Then rRNA, that helps in the formation of peptide bonds. Okay. Question number 7. Which enzyme catalyzes transcription? I mentioned this a couple of times, that the enzyme involved in transcription, what is transcription? DNA to RNA, and the enzyme is RNA polymerase. So RNA synthesis, RNA polymerase. DNA synthesis, which is replication, it is DNA polymerase. So question number 8, pay attention to this question. A DNA molecule has 2 strands, right? Double helical structure. How many strands are used during replication and how many during transcription? So DNA is double helical structure. Yeah. Right. So the double helical structure. In case of replication, each one of the strands, each one of the strands will make new daughter cells, daughter DNA. So each one of the strands will synthesize a daughter strand, right? So this is double helical. Strand number, let's say red strand. Red strand will give rise to purple strand, and then purple will give rise to red strand. Then again, so these 2 strands, so let me tell you again. So what happens that during replication, each one of these strands will separate. So let's say this strand will separate from the purple strand. So there will be a purple strand, they will separate, and then there will be so strands will separate, right? They separated, these 2 double helical strands, they are separate. And then each one of this strand will synthesize a partner for itself. Right? So what is the end result? You get 2 copies of and each daughter cell receives this duplicated DNA. Now, in case of transcription, what happens that only one strand will make RNA. Here, both the strands, but here, only one strand. Whether it is red or purple, only one strand. So if the purple strand is being used as a template, then if purple, then a red strand will be synthesized. If, let's say, a red strand is being used for synthesizing new RNA molecule, then a purple strand will be synthesized. So the so the bottom line is, the take-home message is that, so DNA has double helix. In replication, each one of the strand will give rise to a new molecule. So 2 new molecules of DNA are formed. But in case of transcription, only one RNA molecule is formed. If purple is used as a template, then red, and if red is used as a template, then purple strand is formed. Okay. So let's look at the next question. What are the difference between transcription and translation occurring in prokaryotes and eukaryotes? So I mentioned, I started my lecture with this differentiation, highlighting this difference. So transcription in prokaryotes occurs in cytosol. Transcription in eukaryotes happens in nucleus. Translation in prokaryotes is in cytosol. Translation in eukaryotes is also in cytosol. So to summarize, transcription is depending upon presence or absence of nucleus. Translation in both prokaryotes and eukaryotes translation happens in cytosol. Why? Because the ribosome is present in the cytosol. Now the question comes, 10th, what is the relationship between mRNA and genetic code? Now, remember, I mentioned to you in one of the earlier slides is that I mentioned to you that in the earlier slide that mRNA serves as a template for amino acid, right? So let's say if this is an mRNA molecule, and an mRNA could be, just for sake of understanding, let's say AUG, GGG, CCC, UUU. So let's say this is the sequence of ribonucleotides in the mRNA. Now the amino acids that will come for each one of these triple nucleotide is like this. And this is conserved that AUG, in case of AUG, the amino acid will be methionine. In AUG, the amino acid will be always methionine. In GGG, always the amino acid will be glycine. For CCC, the amino acid will be proline. For triple U, it will be phenylalanine. So who does this? From where do we know that AUG for met, triple G for glycine, triple C for proline, and triple U for phenylalanine? How do we know that? We know that because there is a scientific query, investigation, and that scientific investigation revealed that there is a code called as genetic code. And this genetic code decide which triplet nucleotide will make which, or code for which amino acid. So let me go to the PowerPoint slides. So can you see this slide?
Slide. Yes, slide.
So,
yeah, I see.
So this is a table called as genetic code table. Here, if you see, I don't want to go into the details. So these are triple nucleotides, UUU. If they come together, it codes for a single amino acid, phenylalanine. If it, for UA, so these are various, this is permutation and combinations. And this table that indicates what triplet nucleotide will give rise to which amino acid. And this table is called as genetic code table. Okay. So the question was, what is the relationship? What is the relationship between mRNA and genetic code? So the relationship is that genetic code tells or reveals the amino acid that correspond to triplet ribonucleotides. So each triplet ribonucleotides on mRNA codes for single amino acid. So that's the relationship. Now here are some examples. So let's see. So these I have given here triplet codes, CAG. So CAG, it makes what amino acid? It codes for rather which amino acid? CAG. So if you go here and look for CAG, so CAG is here, CAG. CAG is here. And this CAG codes for amino acid GLN. GLN stands for amino acid glutamine. Okay. So, and another example on the question was GAG. So GAG stands for amino acid GLU. GLU stands for glutamic acid. So these three nucleotides code for single amino acid. So let's look at the next question. So CAC stands for proline. So these triple nucleotides, now I would like to pay emphasis on three nucleotides here. UGA, UAA, UAG. So let's say this is the mRNA molecule. This is an mRNA molecule. Now in this mRNA molecule, 90% of the time, 99% of the time, 99.9% of the time, the starting triplet nucleotide will be AUG. So if there is so cell is very smart in terms of, for example, you're writing a term paper. What is term paper? If you break down term paper, it is collection of sentences. If you don't use a period or a full stop, then you don't know where, you know the starting point, but we can't make out any meaning. So when you convert your term papers into series of paragraphs, series of subtopics, give it a structure, there's an abstract, conclusion, introduction. So you're giving it a structure. So there's a starting point. Okay, so I know that this is a sentence, and then next sentence, then a paragraph, then the second paragraph. So cells are also very organized. If there is a protein, then there is a starting amino acid, and then there is an ending amino acid. So how does how does this end happen? So there is a coding flag sequence that says, okay, protein cannot be synthesized beyond this site. So protein synthesis or protein is not made up of unlimited amino acid. There is a restricted number of amino acids within a protein. Okay. So there is a when a translation is happening, when there is an mRNA strand, and then there is tRNA that brings amino acid from the cytosol. It brings amino acids here. So which amino acid will it bring for AUG? It will bring amino acid methionine. It will bring amino acid methionine. And this methionine will come here. And then let's say a second tRNA comes, it brings amino acid, second amino acid, let's say glycine. So they come together. So all these amino acids, they come are brought by tRNA and they come here depending upon the sequence of mRNA. Now this has to stop. So even there's a stop signal. So what are those stop signals? So I just mentioned in the question, UAA, UAG, and UGA. All these three nucleotides, three triplet nucleotides, they serve as a stop codon. So a protein, when a translation is occurring on the surface of ribosome, the starting point of any mRNA will be AUG that will code for amino acid methionine. And then after some time, when the protein is synthesized, the process has to stop. Just like, we are driving in a block, we see a red light and we have to stop there. If we don't, then we have to pay the penalty or something else could happen. So it has to stop. This red traffic light or stop codons are three: UAA, UAG, and UGA. So remember, there are three stop codons. So that's what it was, UGA. How many start and how many stop codons are there? So I just mentioned to you that in 99.9% of the cases, the starting codon is AUG. AUG is called as start codon, right? This is only one. There's only one start codon, and that is called as AUG. But there are three stop codons. Stop codons are three in number: UAA, UAG, and UGA. What's the next question? Yeah. So this is, if the DNA strand is written as this AATT, so what will the second strand? So let me copy paste this. I don't think I can paste it on the Google slide, so I'll do it here, right? So if I'm increasing the font size, so if this is the sequence of DNA, then on one strand, if there is a DNA on one strand, this is the sequence, right? Then if this is the first strand, then question I'm asking you, what will be the sequence of nucleotide on the second strand? So for A, the second strand will have T. For A, T. For T, A. Again A. For G, C. Again C. G. G. C. A. T. A. So if this is the first strand, if this is one of the strand, if this is the sequence of nucleotide on the first strand, the second strand, the sequence will be like this, TTA. Now, same thing, if let's say if the sequence of DNA so the question number 14th is, if the sequence of DNA is AA, if this is the sequence of DNA, then what will be the sequence of RNA? In other words, if this is the sequence of DNA that will make RNA, then what will be the corresponding RNA? The corresponding RNA would be, for A, RNA will have U, A, C, G, G, C, A, C, A, and then T. So there is a relationship, right? We know that A, nucleotide A, it binds to with double bond with T. And nucleotide G binds with C by a triple nucleotide, right? So there's a relationship. This relationship is called as complementarity. Okay, so with this we finish the review of chapter 3rd, and if you have any question, then please ask me. Do you have any questions?
Professor.
Yes.
My question is, the way the question was set up, you want us to answer it as a DNA replication, or you just want us to answer as a sample replication? Because you didn't put DNA replication, you just put replication.
So replication is always DNA to DNA.
Okay.
No, there's no other form of replication.
Okay, thank you.
Any other question? So if there are no questions, then let's move on to chapter number 4. Chapter number 4 is on genetics, and the first question, or in your homework or class assignment, it's question number 15th. So 15th is what is phenotype, and 16th is what is genotype. Give examples. So I will go to the slides on genetics. Right. So I hope you can see the slide. So let's look into these terminologies. They are the major questions on the assignments. So what is phenotype? Phenotype is external physical appearance of a living things. So for example, in case of plants, it could be, so plants could be, you can look at the height of the plant. It could be tall, it could be short. You can look at the color of the flower. You can look at the color patterns of the leaves. So whatever you can appreciate by observing through your eyes, Those are the physical attributes. The color, the height, the pattern of veins on the leaves, the seed, that if you're buying a seed from a farmer shop, then all these are physical attributes that you can appreciate through your eyes. You don't know the genetics about it. It's just the physical appearance of a living being in case of plants. The next question is genotype. So let's say there is a purple color flower, and this purple color flower is represented by 2 chromosomes diploid by big B and big B, or let's say big P and big P, purple. Then this is the genotype. So let's say the tallness could be, the tallness of a plant could be represented by big T, big T. So this tallness becomes a physical attribute called as phenotype, and the big T becomes the genotype. It represents the chromosome. So let's say if a plant is short or dwarf, this could be represented by small t, small t. So shortness or short plant becomes the phenotype, and a small t becomes the genotype. So let's context of flower. Let's say flower is purple color. So the purple color is phenotype and the genotype is big P, big P. And white flower could be small p or small p. So each genotype, each gene can give rise to, the message here is that each genotype can give rise to a phenotype. So small t, when a plant has a gene or chromosome, small t, then the plant is short. When it's big T, then it is tall. Okay? So that's phenotype and genotype. What is an allele? So allele, so on the chromosome, so this is the chromosome. Let's say this is a chromosome of an individual, a human being. 2 chromosomes, one from father, one from mother. And so let's say on these chromosomes, there are various genes. So these genes are located at a particular position on the chromosome. So let's say hair color. So let's say father might have, let's say, black color. Let's say mom has brown color, right? Let's say a father is short and mom is tall, the growth genes. So when genes are present, so genes are, to sum this up, genes are present on chromosomes at a specific positions, and these genes, they represent characters, right? And these characters could be same, it could be identical, or it could be different. So these genes which exhibit or which codes for or which express similar or contrasting characters. So similar could be both the parents could be tall, or it could be contrasting, that one parent is tall and the other is short. So this, when a gene codes for a character which could be recognized, that gene is called as allele for height, because we are looking into tallness and shortness. So that was question number 3. what is the difference between an allele and a chromosome? So an allele, as I mentioned to you just now, an allele is present on a chromosome. So let's say here, genes and alleles, they are present on chromosome. So the next question is why Gregor Mendel is called as father of genetics. Father of genetics. So Gregor Mendel did all the work to explain the mechanism of inheritance, major work. And at that time, in the mid 1800s, there were less sophisticated microscope, there was least technology, but just because of his observations on the inheritance of characters, inheritance of characters in pea plants. In pea plants, he could propose how characters are inherited from one generation to the next generation. So based upon the flower color, height of the plants, he could understand the mechanism of inheritance, and he proposed the laws of inheritance. But why he became father is because his work, which he did on the pea plant, was so strong, was so solid, that we can explain some of the genetic diseases in humans also. For example, sex-linked disease like color blindness, which is a major problem. If a person is colorblind, he cannot distinguish between different colors. If a person is night blind, he cannot see in the night. So these are inherited through sex-linked. Why I call this sex-linked? Because these, so remember this human beings, we have 23 pairs of chromosomes or 46 chromosomes. The 23rd number chromosome is called as sex chromosome. Sex chromosome. If there is a problem in this chromosome, then this is passed on to the next generation. So if there is any problem in the sex chromosome to 23rd chromosome, and that is passed on from one generation to the next, then it is a sex-linked disease such as color blindness. So that was the contribution of Mendel, huge. He is the starting point for the field of genetics. Whenever you hear the word genetics, it means it is talking about inheritance. Why did the question number 6 is why did Mendel choose pea plants to understand traits? So Mendel did a lot of hard work, but he was very lucky. He was lucky in the sense that he was not observing in humans, because humans, you can only observe for one generation because we tend to live longer. But pea plants, you can grow every year or more than once in a year, and also so you can grow annually at least every year, and you can generate and you can grow. We can grow pea plants or any plant in large number. We can grow pea plant in large numbers. That is good, right? Because we can observe more. We can come up with statistics. We can come up with number. In humans, if you do the same experiment, you can observe one family, and one family could live, depending upon various situations. So, but in 150 years, you can study 50 generations of pea plants or maybe more. And each generation could be thousands of pea plants. So he was a mathematician, a very good observer. He was a very good observer. He loved nature and he was a mathematician. So he observed the pattern of inheritance from one generation of one pea plant to the next. And then based upon his mathematical models or interpretation, he could propose hypotheses. And those hypotheses stood the test of time that in today's date, it's called as laws of Mendel's. Mendel's law 1, law of dominance, law of independent assortment, law of segregations. So he did amazing work for us, especially to explain many human diseases. So that's why he's father of genetics. Okay, so that's why he chose pea plant, because it could grow in large number. He could get a lot of data from them. He can come to a conclusion. So what is the difference between recessive character? What is the difference between a dominant and a recessive character? So let's say a plant is tall, right? So this tall plant we could represent by, so tallness is phenotype. The genotype would be, let's say, big T, big T. Let's say it is crossed with a short plant. Then shortness is a phenotype. This phenotype can be represented by genotype, small t, right? Now, for sake of understanding, let me redo it. So tall represented by big T, big T. Okay. And a short plant represented by small t, small t. Small t, small t. Now, for sake of understanding, let's say, let's give a number to this. Let's say this tall plant is 100 centimeters long, and the short plant is, in height, we could measure, it is 50 centimeters long, right? So if these two plants, they are crossing, they are fertilizing, right? They are fertilizing. So you have fertilizing means male gametes and female gametes. So remember, before fertilization, so a gamete is formed by meiosis. So when a meiosis will occur, there will be reduction in the chromosome number. So reduction will be half. So this will become big T and this will become small t. So reduction by half. Now when these two gametes, when they cross, pollen grain and ovary, when they cross, it will give rise to plant with genotype, big T and small t, right? So tall and short plant, big T and small t, they underwent meiosis to give rise to gametes. This is haploid, half the number of chromosomes, they form TT. Now, a common sense would say that, okay, one plant was, common sense would say that, okay, this plant was 100 centimeter, this plant was 50 centimeter. So they are fertilizing, they have undergone reproduction. So this plant should be 75, right? Average, 100 plus 50 divided by 2, 75. So this plant should be intermediate, 75. But what happens? This plant, it is not 75. It is not 75. Instead, this plant, all the plants are 100 centimeter long. What does it mean? This means that this character is dominating over this character. So tallness is dominating over shortness. So remember, many students, they get confused that this plant is tall here instead of being intermediate is because the short plant is disappeared, is destroyed. No. When one character expresses more over the other, so tallness is expressing more over shortness. So tallness becomes dominant. This doesn't mean that the short character is deleted, is removed. It is suppressed for time being. So separation in genetic term, we use recessive. So tallness is a dominant character because it expresses itself, and shortness is a recessive character because it doesn't express itself. It might express later on. So sometimes you can see that families with shorter parents may have long kids or vice versa. So when you do the pedigree analysis, you find, oh, grandmother or grandfather was tall. So somehow the parents were short, but again, the grandkids, they become tall. So these things, that's what makes genetics very interesting subject, right? It doesn't get over with pea plants, so there are so many attributes to genetics that we still don't understand. We don't understand our own chromosome, right? So a character that expresses itself is called as dominant, and the character that is hidden, we don't use the word hidden. So every area or discipline has its own terminologies. We use the word recessive. So what's the next question? Is what is the difference between homozygous and heterozygous trait? So let's look at this slide. So let's look at this slide. So let's say this is a purple color flower. This is a purple color flower, and right. So this is a purple color flower, and right. So this is a purple color flower. This is represented by a genotype, big B and big B. Since both these chromosomes are identical, so they are called as homozygous. So since the purple color is dominant color, so it is called as homozygous dominant. Now, as I mentioned the word dominant, purple color, so even if big B and small b is present, where small b is a recessive character, white flower is hidden, it is suppressed, it is recessive. So there could be three situations, right? Big B, purple color flower, since these two chromosomes are identical, it's called as homozygous dominant. Big B and small b, they are not identical. Big B stands for tallness and small b stands for shortness. So this is heterozygous. And when small b, they come together, it becomes homozygous recessive. So let's understand in context of tallness. So TT, it represents tallness, and this is homozygous dominant. Small t represents shortness. Because they're identical, so it is homozygous recessive. It doesn't express itself. What about this? So this is big T small t is heterozygous. But because tallness is dominant, all the plants are tall, so this is heterozygous condition. Okay. So just to write here, big T or big P, they are dominant, homozygous dominant. Big T small t or big P small p, it is heterozygous. Small p and small t, they are homozygous recessive. So this is homozygous dominant because tall color and purple color are dominant. Big T and small t, they are heterozygous, and this is small p for white color flower, small t for short flower. They are recessive, so they are called as homozygous recessive. What's the next question is, why did the F1 generation show all the flowers to be purple than a mix of white and purple? So let's go back to slides. So this is from this slide. So in F1 generation, why all the plants are purple? That's because the purple color is dominant. So a color that is dominant will express itself. So that's the reason that purple color is dominant. What's the next question? During complete dominance, what are the genotypes and the phenotypes? So what are the genotypes and the phenotypes here? Pay attention. So what do you see here? You see here, phenotype means visually, physical appearance. So what do you see here as a visual? So there are four squares here, and the 4 squares has 3 purple, 3. So I have 3 purple and 1 white flower in this F2 generation. So the ratio is 3:1, phenotype, physical. But if you look at the genotype, what do you see here? Big P, what do we see here? We see here that big P, there is only 1. Big P, small p, there are 2. And small p, there are 1. So the genotypic ratio is 1:2 to 1. And the phenotypic ratio is 3:1. So 3 purple flower, 1 white flower. 3:1, 1:2 to 1 is the genotype. 1 homozygous dominant, 1 homozygous recessive is small p, and 1 heterozygous, 2 heterozygous. 2 heterozygous, 1 homozygous dominant, and 1 homozygous recessive. So that's so let's look at another next question. Incomplete dominance. Incomplete dominance is, so when there are rules, there is always an exceptions, right? So when Mendel came out with this, many scientists, they try to look at various other situations. Here, if you see, it is called as, so purple flower is a situation of complete dominance. Here you see the white flower and the red flower. When they're crossed, they make pink flower, which means that none of the colors are dominant, right? None of the colors are dominant. So there's blending. So what is the phenotype here? The phenotype here is that there is 1 red flower. There is 1 red flower. There are 2 pink flowers, and there is 1 white flower, 1. So the phenotype is 1:1. Now let's look at the genotype. So genotype, you see that there are 2 CRCRs, right? It's 2 CRCR. Right? There are 2 CRCRs, right? CRCR stands for red, and that is only 1. If we look at the heterozygous, what is a heterozygous? A red color flower and a white color flower. That is CRCW. That is how many? 2. So you see here, CRCW are the pink flowers. So there are 2. And then CW, they are again 1. Right? So in case of incomplete, why incomplete? Because none of the characters are dominant here. So the phenotype in F2 generation is 1 to 2 to 1, and the genotype is 1 to 2 to 1. So phenotype and genotypes are similar in case of incomplete dominance. So let's look at, I believe, yeah. So this is very important questions. First was, give few examples of sex-linked diseases. So like color blindness, night blindness. And the second was why females are couriers. A female can, so let's say in males, right, we have sex chromosome as X and Y, right? So X and Y. So let's say if the X chromosome has a problem, then this male will always exhibit the disease, will always exhibit a disease, right? But in case of females, ladies, what happens that there is 2 X chromosome, right? So if one of the chromosome has problem, then no big problem. Why? Because the normal, so one has problem and one is okay, right? This is okay. So the good healthy chromosome will take care of this problematic X. So this female will be carrier of the disease, but she will be normal. She will be normal, no problem, right? But a male, when X comes, why? Because males have X, only one X. If that is problem, male will 100% will have disease. But a female will have disease only when both the chromosomes are having problem. So when both the chromosomes are having problems in ladies, then only ladies will be showing the disease. That could be color blindness or anything, any other disease also, hemophilia or sickle cell anemia or cystic fibrosis, right? So there are many sex-linked diseases. So female have an advantage because of an extra X chromosome. If it is normal, female could be only carrier. Now thing is that marriage could be, it could be between normal male and normal female. It's perfectly normal. It could be with a disease male with a carrier lady, carrier of a disease, or both could be having disease. So there are various permutation and combination. And then it will depend upon whether a girl would be sick or whether a boy was born to these parents. So all these we could answer by through genetics, and it all falls back to Mendel's work. So that's why Mendel is called as father of genetics. Okay. So we take care of second chapter number 4. Any questions?
No, Professor, no questions.
Okay. Nene, any question?
No question.
Okay. So let's.
I have a question, Professor. I don't have 5.
Say again?
I said the document I have, it doesn't have chapter 5. It's
Yes, yes. So I'm going to read the questions for 5, okay? And I'll upload that. Okay?
Okay.
So so the question, but you have the chapter, but you should have the slides and the video, right? Recordings, right?
Yes.
Okay. So the first question is, what is a plasmid? First question is, what is a plasmid? So in a bacteria, if this is a bacterial cell, then first there is a major chromosome, main chromosome. This main chromosome is called as, nucleoid, right? But these bacteria, in addition to this major chromosome, they also have a small double-stranded chromosomal structure, and this is called as plasmid. So now here, a bacteria will not survive. A bacteria will not survive without this chromosome. If main chromosome is not there, bacteria will die. But if, let's say, if the plasmid is not there, bacteria will still survive. So this is the major chromosome. Now, what is interesting is that both these chromosomes, they are independent of each other. They are independent. They are not dependent. Why? Because they can replicate. So one of the indicator for independence is growth. So this main DNA or main chromosome and plasmid, they can replicate independent of each other. Now, what is this chapter? This chapter is called as recombinant DNA technology. Now, what scientists have done, bacteria can cause diseases, but scientists have converted bacteria into a depot, a warehouse of tools. And that tools they use to manufacture biotechnological products. For example, insulin hormones, right? So insulin hormones, if they want to make at industrial scale, at large scale, we cannot make it in our body. We cannot use human beings as tool. We have to take help. So scientists, they take help from bacteria. So they don't ask help. They convert bacteria into an enemy that could cause disease into a laboratory reagents. So one of the laboratory reagent that bacteria has is plasmid. The second component that bacteria has is called as restriction enzymes. So what happens that in a bacteria, they multiply very fast. So when the bacteria is multiplying, and if we have insulin gene in the bacteria, it will make tons of insulin. So a smart company, a smart biotech industry can harvest those insulin, package them, and then supply globally. And when they supply globally, they help the society, they employ people, they change the socioeconomic structure of the city, of the state. So in one of the major hubs of biotech industries are Boston and California, San Diego, LA, and San Francisco. So they provide a lot of job opportunities to the neighborhood, and it uplifts everything as well as you cater to the society. So now the question comes, how will you take human insulin gene and put it into the plasmid? So let's go back to the slides. So bacteria has enzymes that can cut DNA. These enzymes are called as restriction enzymes. Restriction enzymes are the enzymes that can cut DNA. But the important thing here is that they cut at a very specific place. So for example, let's take an example here. So let's say there is a restriction enzyme. There's a bacteria called as E. coli. And this bacteria makes an restriction enzyme EcoR1. This bacteria makes an enzyme EcoR1. Right? This EcoR1, this enzyme, it acts upon a sequence of DNA. That sequence of DNA is called as GAA-TTC. So you know that the DNA is double helical structure. So now pay attention here. If this is one of the strand, then the second strand is CTTAAG. Now pay attention here. So look here, going from this direction to this direction, what is the sequence of letters, words? GAA, sequence of alphabets, GAATTC. And when we go from opposite end, so here also, same, GAATTC. So such sequence are called as palindromes. And they're present in English literature also. I gave an example of words such as madam. So a word madam, you can write M-A, alphabet M-A-D-A-A-M in this direction. And if you go in this direction, it reads the same. Such words are called as palindromes. And why? Because there is a symmetry in the center. And both these letters, they are just like mirror image. So such words are called as palindromes. So you see this palindromic nature in the action of restriction enzymes. So these restriction enzymes, they can cut the DNA at a specific site, palindromic sequence. And when you cut in the plasmid, you can insert insulin gene. So let's take an example. So let's say that I'm drawing a rough structure of chromosome, right? So let's say this is centromere, and let's say this is the one chromatid, second chromatid, and here, so this is a rough structure of chromosome. Now let's say that insulin gene is present here. Now why I chose insulin? Because if insulin is messed up, problematic, a person doesn't have insulin, then the sugar will go up or down, and it can cause, continued abnormal level of sugar can cause, not only diabetes, but other problems. And now you know that diabetes are present in kids because we have changed our lifestyle. So to treat diabetes, we need insulin at industrial level. Industrial level means this insulin should be available at the pharmacies of not only US, but all over the world. So we make huge amount of insulin. Now, can we derive insulin from human? No, that's unethical. So what scientists will have done, they have taken the human insulin gene and they have put the human insulin gene into, they have put the human insulin gene into bacterial plasmid. So this is human insulin gene, into the bacterial plasmid. Now, I told you that bacteria, they grow at magical rate. So if the more food you provide, the more bacteria will grow. So the bacteria, they grow at logarithmic scale. So with time, bacteria will multiply. And when the bacteria is growing and the plasmid is growing inside the bacteria, this plasmid is growing inside the bacteria, then it is producing large amount of insulin. And this then bacteria can be grown in an industry. So what is happening now? Let's understand the same thing in terms of COVID. What is happening in COVID-19? So there is a company called as, if I spell correctly, Moderna, right? This is Moderna. This is leading the efforts, US efforts for producing the vaccine for COVID-19. So they have to make a huge amount for not only US citizens, but all over the world if they want to be a good business, structure, be a good business model. So they have to hire people, they have to make team. So they will employ people, it will generate economy, it will generate revenue, it will improve the city, and then it will help society. So, so they have to make in huge amount. And so they have to take help. You cannot make in humans. Although vaccines will be injected into humans, but humans cannot be used to make vaccine. So they will use some tools, such as bacteria. In bacteria, what they will use plasmids. And then they will make huge amount of, so I don't want to go into the details, but that is the basis for recombinant DNA technology. Recombinant means what? Fusion. So when you fuse, so what the word recombinant means in fusion, right? You have fusion music, here you fuse DNA. Which DNA? Let's say human DNA plus bacterial plasmid. And why do you do that bacterial plasmid? Why do we do that? Is to make a product that can treat human diseases, right? So remember, how did we started this class? We started with atoms, periodic table. Then we went into molecules. Then from molecules, we looked into cell structure. From cell structure, we went to how cells divide. And then into how cells are replicated, transcription, translation, and then we are looking into the applied aspect of this knowledge. So when you apply basic fundamental knowledge to a business model, then you bring technology. So that's why this course is called as biotechnology, using biological principles. And then when technology impacts society, I agree both ways, but then you do mostly helpful. So you have a lot of presentations on GMOs, algal fuel, and you'll see more stem cells, and you will see more. So with this, we end the review of chapters 3, 4, and 5. and you can ask questions. Thank you. Any questions?
No, Professor. Thank you.
Okay, it
No,
was a long,
Professor. Thank you.
long session. So thank you and good luck for midterm. Okay.
Thank you.
Multiple choice questions and 30 to 35 questions.
Okay.
Monday. Okay.
Okay, Professor.
Okay.
Thank you.
Take care. Have a good evening.
Have a good evening, Professor.
Take care.
As well. Bye.